You guys see what I do, yeah? Okay. Good morning, everybody. The last session of lecture style followed along part of the workshop. And for those of you who do not do the homework, this is the last one. So if you have questions, this next 3 hours is the time to raise them if you are not here for the next day and a half. Those of you who are doing the homework, we will start after lunch. Ruben will upload the homework package as a PDF file so you can follow through and the data that you need for it. And then you have this afternoon, as long as you want to work on it, and tomorrow until 8 o'clock to finish the homework and submit it. The instructions should be clear that you will get you the PDF file. As for those of you, so everybody is welcome to look at the tasks, even if you're not doing the homework, and it will benefit if you at some point follow through and try to do the tasks, even if you're not getting the credits for this workshop. Those of you who do it, we will also give you a printed version of the instructions. We made little booklets out of them, they're very cute. We will distribute them after lunch. We would have given or wrote them, but the assistants are not here yet. So you will get them at lunchtime. This is only for those who are staying or those who are doing the actual homework. In terms of doing the homework, there will be 2 sets of questions. One is about microscopy. You have to solve them and pass the test, 50% minimum. And the second one, much harder one, about JIPipe. JIPipe, the JIPipe task will be complex, you will see. But because of the complexity, last year we experienced that the attendees or students who stayed here and worked together in this seminar room, which will be open until tomorrow at 8, did better in terms of more efficiency, because I already see that you work in pairs and triplets, and I love that because it's much easier to share information. And you are very welcome to do that for the homework. Actually, it's recommended, not recommended, but we think it's a good idea, because then you can work together. Plus, Ruben and I will be here the whole time, and we also have lovely colleagues, including Marius, and later another colleague, Drew, who will be here to help in case you get stuck during the homework. Because the idea is that you learn, not that you fail the homework. It is difficult. The instructions themselves, I forget how many pages they are, but 60 pages of instructions.
16?
60. That's because we give you a lot of help. Not as much as you hope, probably, but we get enough. No, seriously, the idea is that you develop during the next day and a half a complete multi-compartment real-life project in JIPipe. And the project which we prepared, it comes from my work. It's from a paper we published like 2 or 3 weeks ago. So it's really real-life, okay, maybe a month ago, real-life example. So you will learn how really to use JIPipe. Because you learned a lot from Ruben. It's fantastic, but we have bits and pieces. And I would like you to get this all together in your head. And my goal today is exactly that. I will go through another project, not the one that's the homework, unfortunately, but another one, which is also a complete project and also literally a few weeks old in terms of manuscript. It's not terribly difficult, but has everything that you need for building a complete workflow, multi-compartments, etc. We will slowly go through this in the next couple of hours. And if you follow through, then you will learn how a project can be built. For this one, if you go to the, if you want to follow through with me, which I recommend, you don't have to, but I recommend, then you can download the project. I will tell you in a second, with the data, and then just follow what I do. Go through the notes as I go through them, so you learn how this is built. You don't have to build the project. That would take way too long. So I built it for you. I ran it last night over here after you guys left. Make sure it runs. And I recommend that you follow and go through it. The data is not too big, so I picked a project which is not heavy on the image size, so you can laptops also follow. Normally we work on much larger PCs, of course, but this you should be able. I also tested it on a small laptop. And then if you follow through and you have questions, my friends are sitting in the back, so just raise your hand and they will help you. You can also ask me why we do it this way or that way. But I recommend that you do follow with me. But you don't have to, if you don't. And to get it, if you go to the shared folder that Ruben and I set up for you, where his data was yesterday, there is now a new zip file for advanced JIPipe. If you download it and unzip it, you will find two projects there. One of them will be called perinuclear. That's where my pointer is at. That's the one you will need. The second one, which is at the top right now, supramolecular, blah, blah, I didn't rename it. This is an example, which is a gift for you. If you have time and if you have questions, you can also look at it, but it's a much more complex one. This is also a published work, and you can use it later to get ideas how certain things are done. To JIPipe, but to follow me, you will need the perinuclear one. Yes.
So from when do we download?
The same page where you
It is now updated via the website.
It's additional data.
No, I will wait 4 minutes until the website updates itself.
Just let me know when you guys go and then it's on.
It's not there yet. It's a minute to update.
Was it the nuclear or supramolecular?
The perinuclear. You are welcome to keep both, but I will talk about the perinuclear. That's a simpler one, but still a good example. The data is small.
So it's on the Google Drive, right?
It is on Google Drive, but Ruben also said he moved it to the website also.
Yeah, it's to the website, but I added the link just now. Because I saw it, and it takes like a few minutes until the website updates itself from the cache. This is the next cache.
So that should be a.
It's on the Google Drive.
And then we need to download advanced. Did you move the zip file or just copy it? Did you move the zip file from the
I just renamed it to make the name.
Oh, just rename it. Okay, that's why I don't see it. What is it called? Advanced workshop data? Yeah, this one. Okay, so if you can, so when you look at the shared drive, there is also a file called templates. It's a JSON file that's on the shared drive given to students. You can also download that. We don't need it today, but that's the templates that Ruben mentioned yesterday. And one of you also talked to that when you have, for example, groups of nodes or complex nodes that you figured out and you don't want to do it again, you can save them as templates and Ruben you showed them how to do that, right, Ruben? Templates.
I explained the concept, didn't show how to save them.
No, that's okay. Okay, and then you can use them later. I will show you at some point how to put them into JIPipe. So what I need you to do then is simply start JIPipe, and then from the folder that you downloaded for the perinuclear in here, I need you to open the perinuclear.jpipe file. And I will do the same here. So we open it, and then this is what you should see. So these are the three compartments that we will work with. These three guys, and this shows you a principle which I would like you to learn and to understand. That basically, you will notice that your code will be much more maintainable if you organize the processing into compartments. And Roman explained how compartments work, but this is a typical simple workflow where in the first compartment, you read in the files, you discover the file structure, which are the folders, which are the subfolders, which are the images. You do filtering for the images because you don't want all of them. Like yesterday, you guys had a trick played on you by having non-TIFF files in the folder, and then if you don't filter them out, then JIPipe will hate you for it. And then in a second compartment, you do your segmentation fields. And the output of that, which is usually either masks or regions of interest, you pass them on to a quantification compartment where you will do your measurements, whatever they are. And this way, when you have to change something, it will become much easier, because if you put all the nodes into one compartment, that becomes really a big mess very, very quickly once you have a decent sized project. But the first thing I need you to do is update the user folders. You guys learned about this yesterday. And if you go to project and project preview, then you will get a tab just like this one. And if you remember this from yesterday, then on the right side, you will find, because you're using my saved file, two preset user paths. And we will need this for both loading the data and for saving the results. And these are for data folder and analysis folder. Now I need you to update them because this one is set up for me. So if you go to select one on either of them, and then you can find your way to the folder where the data is. Now, if I go back here for a second, you will see that what we need is the perinuclear signal folder, and within that, there is a folder called data. That's where I put some example data for you guys. This one here. But your drive may not be called C, and it may not be, it will not be in this folder. So I need you to use the select button to navigate to the folder where your data is. So if you look at my folder structure, and this should be the same in your case, you will have a code folder and you will have a data folder. And you found the JIPipe file in here, this guy here, that's what you loaded. But then I need you now to find the data folder, and when it's in here, in this directory, just use current directory. If you go inside there, then it will also know that now you need blah, blah, blah data. This is what I need you to do. This way the program will know where the data sits, and we do not have to every time. There's a question up here, Marius, maybe? So once you have that, you just click on open. And now you will have the data folder updated. Okay? And then we do the same with the analysis folder. This will be the folder where the results will go. And here you have freedom. You can select any folder that you want. What I did was to select the folder where my example was sitting, and I made a new folder called analysis. So if I go here, I made this folder for you guys, perinuclear signal, and then I made a folder called analysis. I need you to find it. Click on use current directory, and this will dedicate this folder as the target for analysis output. And then you click on open. So now we are ready for the analysis. Let me know when you guys have that. Yeah. Okay, so if we have that, done with the hot part, we can move on. Now let's look at the compartments for tab. And this I already talked to you about. So these are the three folders that three compartments that we will go through step by step in the next couple of hours. So I want you to double click on the file handler. I already have it open, but same thing. So double click. This will take you inside the compartment. And as you see, we have several nodes. It's not too complex, not too many nodes. And again, I would like to emphasize that if you look on the left side, you will see that this is really not a complex compartment. And later you will appreciate that, because when you try to modify your code layer, you will find that it is a lot easier to work with a few nodes, like 10 and 15 at most, than working with 40. And because you have these connections, as Roman told you yesterday, when you have a lot of nodes, this becomes like a bowl of noodles, as you like, or spaghetti. It's very, very difficult to follow. Roman added tools like you can highlight a single connection, you can delete it, etc. So it's much easier than it used to be, but still it's beneficial to keep this relatively simple. So let's go through the nodes one by one. This guy here, if you click on the top one, project user directory, and click on parameters, you will see what we have done here. So basically, the name of it doesn't matter. What matters is this. This is the key that I gave to the folder which I named data folder. And this is, we were just discussing this with Ricardo here, that if you don't remember the key you gave to this data folder, then you cannot use the option. So this has to match the name of the key. So if you remember when we went here on these guys, when you set up the key, for example, we went to data, and I said use current directory. Let's say if I wanted to add a new one, and I use the same one, it will ask me, what should be the key? And this is the one I must have set up as data folder. Yeah, so this had to be named like that. Otherwise, it will not find it. The name can be anything that you want. Okay. So if we go here and we run this node using any of these options, then what you will get in the results table is the location of the folder that you set up. And this way you can check where exactly you are and what the parent folder is and what it is. This is just to double check that you did correctly what you wanted to do. So we are done with this one. Now out of this one will come the location of the folder where the data lives. Now what do we do with it? And if you remember yesterday when Ruben briefly talked about groups, node groups, here I gave you a real life example of a group of nodes. And if you double click on this one, which I recommend, then you will see that what is inside. And we will go through this one by one. So basically, the principle is the following. If you have certain tasks that you carry out in every single project, you don't, and it consists of 5, 6, 7 nodes. You don't necessarily want to have 6 or 7 nodes added to your project every single time you write a new one. You group them. This is only working if you are sure that the nature of the nodes and the number of the nodes is not going to change when I'm using this group. Because silly to make a group and then have to go inside the group and change the content of it. So then what's the point of using a group? So you only make a group if you are sure these 5, 6, 7 nodes I will always use, and I will use them always in this order. Now what did I do here? I did the following. I said, okay, my input is going to be where the folder where the data. Then in this case, we use the project user folder. You can also just drop a folder on your user. But what I always do is I will go and annotate the folder where the data is coming from. So that even though JIPipe knows, because we have a data folder, user folder path, I don't know it. I don't have it as an annotation yet. So I will annotate the folder. We will look inside this in a second. Then I will always check what the subfolders are. Are there subfolders and what are they? And once I have the subfolders, I will always want to remember, because then I may say, here's the data, but I'm now developing my workflow. Please only use one subfolder out of the whatever, 5. Because I don't want to run batch processing right away from the beginning. So I will remember the subfolders, and then I may check again if there's a second layer of subfolders. Maybe it's folder, subfolders, and each subfolder has subfolders. I have given you an example in this project where I check for 2 layers of subfolders, and then I will always do that. And then what do I do after that? Well, once I have the subfolders and I remember them, I will now look inside the subfolders and I check what's in there, the image files, hopefully. And once I have them, I will list them and I will remember their names. I will call them image, for image name. And what do I do after that? Well, like the trick you guys were playing yesterday, I have to filter the findings because maybe there are more than one file type inside the folder. I only want, let's say, the diff files. I don't want CSV files. I don't want XML files. I don't want PNG files. I only want the diff files. So I will always have a filter path here. Maybe I don't fill it in, but I would like to have it. But now I have a folder, a node structure. How many is that? 7 or 8 nodes. Which every single time I read data, I will use. Yes.
Yeah, this was the question. So you can use this individually for every different project. Just saying, okay, this is the data hole and it's find its way.
Exactly. This will find this will make annotations, and the advantage is that the annotations will always be called the same. So you get used to it. And you don't have to look it up. What did I call the image? I called it, I make it important, like you already explained with the hashtag image. What did I call the fold subfolders? I called it hashtag subfolder. You know? So this way, when you code later, you don't have to look it back. It's always called the same. Yeah? And this is a tremendous advantage. Now, Roman explained yesterday how to make a group. So basically, when I developed such a workflow, I highlighted these 7 or 8 nodes, and I said, okay, I want this to be a group, and I want to call the group whatever you want to call it. I called it read, blah, blah, blah. And go back to here. So this is what I call it. Yeah? You call it something that makes sense to you, so you remember what you put inside. And Ruben also explained yesterday how to get some parameters out of it. Now, why do I need that? I need that because the last thing I want is to have a group, and every time I put the group into a workflow, go inside there and change the settings in the nodes. I want to access the settings that I really want to access available without having to double click on the group and go inside it. So in this particular case, the one thing I really cared about, because the rest was constant, I will always call the annotations the same, image, folder, subfolder. The one I really cared about was the filter. How do I tell JIPipe which images to select from the folders and subfolders that I specify? So from this group, there must be a way to access the filter without having to go inside. And you already learned yesterday how to do that. But basically, when you activate this node and you click on parameters, this blah blah you can forget, but this is the important part here. That part here. And take a close look and compare it with the folder that you downloaded. If you look at your data folder, oh, thank you. If you look at the document here. Okay. Oh yeah, it's just so. If you look at the data folder that I prepared for you, you will see that, come over here, that what I do here is going to go into the last node in that group. That node was called filter data path. And what I type here will go into the group and it will be typed into the field of the filter I will show you in a second. And what do I do here? Well, first of all, I say, make sure that you are looking in the data folder. And when you are there, make sure that you only look at the diff files. If you don't do this, and you not forget, but you don't want to remove non-diff files from your data folder, and you try to read, like you guys experienced this yesterday, a CSV file, for example, it's going to yell at you that there is a null pointer error because I don't know what to do with it. It's not an image. This will always be there because sometimes it's not a diff file, sometimes it's an LSM file or a CZI file, but this has to be there just to be sure. Now, what do I call the annotation, which is the file name? Well, because of the group structure, I always call it image. And that's what I mentioned to you earlier, that I didn't have to think about what did I call the annotation which contains the image name. I always call it image. Why? Because inside here, I'm using a group, and that always names it image. Makes it life much easier. But then I prepared for you guys 3 subfolders, and I don't want to spend too much time running the program, so I said, okay, for today it's enough if we go with the, this means donor mail number 6. I only use this folder. So when we test the data, we do test it with multiple images, but we don't test it with all the subfolders, so it doesn't take too long to develop the workflow itself. You don't have to do it. You can immediately start doing the whole data set. I don't recommend it, because at the beginning you will be experimenting with parameters and it's going to take you forever. Actually, what I do recommend is that sometimes you even put a direct filter to the image name. You pick one image that's characteristic, or 2 images which are characteristic. And for example, in this folder, I had images which the biologists prepared, where they simply made a TIFF file, but the TIFF file was a 2-dimensional image instead of 3D, or a multicolor RGB image which showed the overlay of the various components just for us as an information. But I don't want to analyze that. But I also don't want to remove these images from the data folder because this is the complete data set. Maybe I want to look at later the overlay, but I don't want to analyze it. So what I did was I said, please, if the file name, the image name, contains the word overlay, do not analyze it. So I want overlay in quotation marks, not in the image annotation. Ruman showed this yesterday, the negative selection of annotations. This is sometimes much more efficient to tell it what not to do rather than specifying what to do. So this will then go to this folder, look at only TIFF images, only in this subfolder, and it will avoid those images which are just for show and not for analysis. This is very, very important. So when I run this one, it will run everything inside the group. And I wanted to go back for a millisecond here. If you look at the last node and you go to parameters, this guy here contains exactly what I typed into the group parameters tab. So I did not have to go in here and type it in. I typed it in from the main workflow. Is it possible to go in there and type things directly? Of course, it's just not very convenient. So this node is now ready, and we run it. And what we have is on the right side, it will show you all those images which it found. So as you see, these are TIFF files, image 2, 3, 4, 5, etc. And then if you go down to the right, you will see the annotations which are generated. These are all generated inside this group. For example, here, the first one, this remembers the folder. This one remembers the subfolders. This one remembers the second layer of subfolders, if there is one. Sometimes there is only one. And this one I put together to remember the full path, which contains the hard drive name, folders, my name, blah, 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 just in case we need it later. Now, for example, in this case, this checkbox here was crucial because normally you only remember the name of the file or the folder, but here I asked it, okay, please do annotate with the full path and call it full path. You may or you may not need this one later, but I like to have it. Here, as you see, this one is not clicked, yeah? Because I only want to remember the name of the subfolder, not the entire path to it. But if I need it, I have it from this one. And this one just remembered the image name. And like I mentioned, I always call this hashtag image, hashtag folder, hashtag subfolder. So I don't have to think about it. Super. So now we have the files that we wanted to use. If I look at the image, annotation here, you see image 2, blah, 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 blah. Now what are these guys? Well, these are the subfolders. These are not images, so we will not load them. And indeed, if I go to the left side of the cache table, it will say, oh, these are subfolders. So I have my annotated path. I have the files that I want. Now, by the way, here, how these were generated, this is how you handle a group. Now, let me explain that in a little bit. I like the group concept. You see this node here, this is the same as a compartment has. It has an input node and has an output. So when I generated a group out of these 7 or 8 or whatever number of nodes, it automatically gave me an input node to which I connected the user folder data, and it also gives me an output node. And then I said, okay, using the plus sign, I said, please make 2 outputs. I called one annotated paths, and I called the other one subfolder. Because I wanted this group to give me the files which I want to work with, but also the subfolders, just in case I need them. And then what did I do? What I did was I connected the last node after the filtering to the first output. So these are the image names. And here I simply connected the output from the second layer of subfolders. You don't have to do this. You don't need to put out the subfolders if you don't want to, but I do use it time to time, so I like to have it. This is what you really need, the output of the images, so the names of the images that I want to work. So this is something you set up yourself, and just like in a compartment. So what do we do with these images? Well, we read them in. There are 2 ways. There's a read image, and there is the bioformats importer. Ruben probably told you about this. This is a more sophisticated one. So if you look at parameters, you have all these options. Do I want to remember the image title? Do I want to separate the various series if it's a multi-series image? And so on and so forth. This is from ImageJ, the order of the channels. So this is X, Y, and then channel, and then Z, and then time. If it's a multi-series image, I can decide which series to open if I only want to use one or more. And then a lot of other options. Do I want to split the images now or later, and so on and so forth. This is important, auto scale. This ImageJ by default does that when you load an image into ImageJ or Figge, it will scale, so you see it. And sometimes you see this if you have a TIFF image and you load it into Image Viewer on your Windows or Mac, maybe you don't see anything. It's just black. But if you load it into ImageJ or Figge, you see it. That's because of the auto scale option, which is by default on for ImageJ. This can be good, this can be bad, because if it's auto scale, you don't know what the real brightness of. So when you start analyzing your data, unless you look at the histogram of the intensities, you may not realize that this image is way too dark, your signal is way too weak. So again, a plus or minus. I have this on here because when I read in the images, I want to see them, so I can judge what did I load. Do I want to work with this image? Is it good enough, and so on and so forth. But be aware of it, because sometimes also with thresholding, it's a problem if you look at the auto scaled image and it's actually not covering the reality. So let's run it. This should run on your laptops easily because I chose very small files. And then we have in the cache 16 images. You can click on one of them, and then what you will see is that we have two channels, channel 1 and channel 2. This one is the fluorescent signal. This one, not surprisingly, is the nucleus. To see them better, I clicked on this one. So fluorescent signal, nucleus. And the task will be to find the nuclei, to find the cell itself, and then measure the fluorescence outside the nucleus in the entire cell, so everywhere except here. And hence the name of the project, the signal in the perinuclear area, which is in my case a 10 pixel wide ring that's outside the nucleus, directly next to the nucleus but outside of it. This is the perinuclear area. These are 2D images because they are just faster and smaller, so you can easily work with them, but the same principle would apply. So what do we need to do? We need to find the cells based on this channel. Then we need to find where the nucleus is, not based on this dark spot, but the labeling of the nucleus. I think that is order. And then using the nuclei from the nuclear channel, we have to find this 10 pixel wide region which surrounds the nucleus. And we do this per image, and we only use the largest cell in the image because sometimes they have more than one cell in the view, but the other cells are not complete. So we have to find the cells. If there is more than one, pick the biggest one, find the nuclei, find the perinuclei, and measure the fluorescence in the cytoplasm, that is excluding the nucleus, in the cytoplasm excluding the nucleus and the perinuclear, and the perinuclear. It's really an everyday task that biologists will have to face time to time. Now, how do we start the analysis? We have to separate the channels. But when we separate the channels, we assume there is channel 1, fluorescence, channel 2, nucleus. So there is a problem here. This is frame. JIPipe thinks that this is a time series. It is the way it was saved from the microscope or converted into TIFF. Unfortunate, because these are not time codes, these are channels, and I need them to be channels. So do I then just remember that these are channels, and every time I face this question in the workflow, I correct it? No. What I want to do is convert the time dimension into channel. So flip that. Trying to change that. And this is what I'm going to do in the next node. So I'm going to close this one. And that's what I'm going to do here. The node is called reorder dimensions. And it does exactly that. And this comes up surprisingly often. this guy here, and this is what you do with it. You say, what is time, what you think is time is actually channel, and what you think is channel or should be time. The Z, if we had Z, we are not going to touch because that's correct. For these two, I have to switch. So what you do is, now it is set up. But what I did to set this up was, when I said move channel to time, I went here, and the default was, of course, channel to channel assignment. But I said, nah, this should be frame. And what you think is frame should actually be channel. And this, don't touch this one. Okay? So now what we did was we flipped time and channel. So when we run it and we open one of these guys, now it knows that this is channel. This is channel 1, and this is channel 2. Nucleus versus intensity of the fluorescence. So now we are okay. This you don't always need, of course, but you must check when you read in your images, is your order in the dimensionality correct? So we have our images, we read them in, but there are a few properties that we are going to have to remember. So I'm going to annotate, you guys saw this node yesterday. I'm going to annotate my data with image properties. Let's click on this, and then let's click on parameters so I can explain what I'm going to need. Well, first of all, the most important part from here is going to be that. How many channels do I have? If I have time frames, like you are tracking immune cells moving around in a time series, you should know how many time frames there are. In this particular project, we won't need it, but I clicked it on so I can talk about it a little bit. This will be important. What is your X, Y, and Z pixel and voxel size? How high and how wide is the image? And how many Z layers there are, if there are any. You can choose all of them, of course, but you don't have to. But what we will really, you will see it in very shortly. Let me make this a bit bigger. What we will need is this parameter very soon, and we will need the number of channels. Now, JIPipe will give these guys a default name. The number of channels in JIPipe is called image composite channel count. This tab here means this is an annotation, and when I click this on, it will add this annotation to my data set. If you want this to be important, you would have to hashtag it. You can change these. So this is, as you see, this is editable. It's entirely up to you. You can call the number of channels as number of channels. It doesn't matter. If you call it this, you just have to remember how it is called. I usually don't change them because then between projects, I can reuse my nodes because the name of the annotation will be the same between various projects. Okay, so when we run this guy, we will have a whole bunch more annotations. So when I look at the cache, in addition to what I used to have, now we have all these new annotations. Beautiful. With this one, you have to be a little bit careful because it's very easy to get carried away and annotate everything. And then eventually, when you try to match images based on annotations, this can be a pain in the neck that you have too many of them. But what you need, you need. So I follow the policy that if I need an annotation, I pick it up early and I carry it through the workflow. So all the sub-steps that I will put up later, they all know what the image size is, they all know what the image resolution is, rather than having to edit later. And then you have a problem that inputs which were done before you added the annotation do not know, let's say, the image size, and the new ones do, and now I'm doing. It's getting very windy. Wonderful. So now we have all these new annotations. and we are very happy about it. This one, this guy here, that's the resolution. That's the pixel size, 54 nanometers. So now, without asking the biologist, I already know what the resolution was. This is a high resolution microscopy, STAT microscopy data. So, why do I need that? Well, you will see it in a second, but first of all, so we ran that, we go to the next node. I added an annotation editing node here, set edit annotations. And let me explain you why I did that. So we try to remember everything that we will need daily. Folder name, subfolder 1, subfolder 2, image name, etc. Now, here I introduce a second image name, which is a combination of the subfolder within which the image lives and the name of the image. And I put an underscore so they don't go, blend in together. Why did I do that? Well, the experimentalist had the unfortunate habit of not naming the images, but just letting the microscope name it. And the microscope named it image 1, image 2, image 3, image 4. So you have like 5 image 4s, 3 image 10s. So the image name itself doesn't tell me anything about who was the donor, what was the experimental conditions, nothing. It's just called image 1. But at least they put the data into subfolders where the subfolder name is indicative of the condition, for example, which donor it came from or what chemicals or conditions were added to the data. So in order to be later, to be able to separate the results into groups which correspond to the conditions or the donors, I had to have a new annotation, which is a combination of the condition and the actual name of the image. So this will be like, for example, DM6, male donor 6 underscore image 3, image 4, etc. Now I know if I use, but for this one I needed a new annotation. And I call it image 2 because I don't have too much fantasy of naming it. But now I know that image 2 is a more complete information carrier than image itself. And this shows the principle I mentioned that if I need an annotation throughout the project, I add the annotation as early as possible. I don't wait until later. Now let's move on. So this was a bit of a sloppy data set, so I chose this also for this reason, so you know how to deal with that. So when I run this one, I will have a new annotation, image 2, and indeed it's called the donor plus the image name. So now I know this is male donor 6, image 15, so now I exactly know which image I'm talking about. Now comes a very interesting step, this guy here. The data set which I got, again, was a bit sloppy, so they mixed the following things. And I could have just deleted the things I don't need, but I didn't want to go through them and one by one. First of all, they gave me images which had more than 2 channels or only had 1 channel. Some days they decided, no, we're not going to record the nucleus. The data is useless, because I cannot find the nucleus if I don't know where it is. Some days they had the 3rd channel, which was useless. But if I have a 3rd channel, then I have to tell JIPipe, okay, what do I do if it's 2 or 3 channels? So first of all, I added a filter that said, if the image composite channel count is 2, then keep the data. If it's anything else, do not. So this way, the images they gave me, which had only 1 channel or 3 channels, will not be considered. So I don't have to manually remove them. Why do I call it this? I call it this because that's what the previous node, where we annotated with image properties, called the number of channels. And like I said then, I could have renamed this to number of channels, but I didn't. I just had to remember what it's called. Now you will see this funny thing here. I don't know if you guys talked about it yesterday. if the name has gaps in it, spaces in it, then putting it in quotation marks is not enough. You have to precede it with the dollar sign for JIPipe to understand. If you don't remember this, and you go to the edit option, Roman explained this very well yesterday, and you type this in, that they say, okay, this is the name of the annotation. How do I quote this? Then the edit function will put this in quotation marks, and it will put the dollar sign in front of it. Eventually, you will remember this, so you don't have to, but edit will always happen. Now, the second thing we had to do was the color. They gave me data which was at high resolution, like the one you saw, but they mixed it with overview images where the resolution, instead of being 54 nanometers, was 200 and something. Confocal image instead of a static. And I asked, okay, do I have to analyze these? Because the resolution is not really there. They said, no, we don't have to have that. We just analyze those, which is at static resolution. But then I had to limit my analysis to those images where the resolution is high. So what did I do? I did grab the annotation which I added a couple of nodes ago, the pixel size. And the pixels are square, so it was enough to grab either the X or the Y, so I picked the Y. And this was called by JIPipe physical dimension Y. Again, the dollar sign, because it has spaces in it. And I said, okay, this is a string. What I need to have is the resolution. So I need to have the first 3 or 4 digits. 0.01, that was 0.05, that's the high resolution, 0.2, that's the low resolution. So I said, okay. And Ruben explained this yesterday, how this substring, this is a string manipulation. You take the annotation and you say, between the first one and the fourth one, I need the substring, convert this to a number, and check whether this is below 0.9. If it is, then it's the 0.05 image. If it's not, then it's the 0.2 image. I don't want it. So I say, okay, only keep it if the resolution is high. So this way, I don't have to go through the data and manually remove the low resolution ones. And if you can imagine, if you have thousands of images, you really don't want to do that. And neither does the biologist. So you do this via JIPipe. So now, if I run this one, I will only have 10 images. If you guys remember, here we had 16 images. So 6 of them were either more or less than 2 channels, or they were low resolution images. But after this node, I only have 10. So now I have the images that I really want to work with. So what do I do with them? Well, we split the channels, as you guys were explained yesterday. Now I have 20 images, 10 of them for the nuclei, like these guys. You probably heard this yesterday. You can scroll through the various images here with this next and previous button. Control arrow will do the same. So the nuclei are quite nice. This image has 2 of them. The others have just 1. And we also have the fluorescent labels. So now we have them for each of the 10 cells. So these are all now high resolution images. So we have what we want. I also like to color them, but you don't have to. So I used the set lookup table color map option. I set the nuclear image to cyan, and I set the fluorescence to yellow. I like to work with the cyan, yellow, magenta triplet rather than red, green, blue for people who don't have an easy time separating RGB colors. So the colorblind or limited color perception. And then I put them out. Now, how did I know which channel is which? Well, this is the node which does that, split channels. And if you go into that node, you will see, first of all, that it has a plus sign. And since I know from this node, for the filter annotation, that I'm only going to have images which have 2 channels, I could set this split channel up, hard-code it for 2 channels, and I said, add an output, call it nucleus, add the second output, call it label. Then when I go to parameters, I have now 2 channel assignments, nucleus and label, but I still have to tell JIPipe which one is which. And I remember from up here, if I open one of these guys, that the first channel, channel 0, was the fluorescent signal, and the second one, this is channel 1, was the nucleus. So when I go back here, I can say nucleus is the second channel, label is the first channel, but counting start at 0, as Ruben explained yesterday. So this is 0, this is 1. You can use the arrows to increase these. So when I run this one, it will split the channels, and then I set the colors up. And then from the compartment, I will let these guys go out via the output channel. And as you were explained yesterday, it's easiest if you grab the node output that you want to output and connect it to the plus sign, because then it knows exactly what type it is. For example, it knows that this is a grayscale image. Otherwise, you have to scroll through dozens of options and you make a mistake. So we have an output, we run it, and what we get is naturally the nuclear images, 10 of them, and the fluorescence images, also 10 of them. So I go back to compartments, and you see that we are here now. I named the output channels, but you can name it whatever you want. And now the question is, what do we do with it? Well, now we have the channels separated. We have the images selected, so let's start to segment them. So we go to the segmentation compartment. So let's go in there. And then let's decide what we want to do with these signals. Now, first of all, the segmentation for the areas that I want will depend on whether I'm working with the nuclei, or which is from the second channel, or whether I'm working with the fluorescent signal which is coming from the first channel, channel 0. So I separate my analysis or segmentation based on the outputs. So on the one hand, I have to find the nucleus. And here I chose a way which uses artificial intelligence. It's not entirely necessary, but it does work very efficiently and very well. And the reason I did that is that if you segment nuclei, the labeling is never entirely homogeneous. It labels DNA, so it reflects the DNA-RNA ratio. You can find it if the contrast of the image is high enough based on intensity. And then you just make sure that the output is smooth. You will see how we do that. Or you can say, okay, these are nuclei, so a trained model will probably recognize them. So let's see how we do that. And this is then going to be this part. So first of all, I like to work with images where I normalize the intensity to 1. This is because, like I mentioned earlier, the ImageJ has this option to have auto scale or not. And if you auto scale, you're always dealing with images which are from 0 to 55 on your display. But when you don't auto scale and you select your, let's say, thresholding method, if a second batch of images or a second replicate, for example, or a second donor was recorded at lower intensity, the same segmentation methods may or may not work because they may or may not be sensitive to absolute intensity. So what do we do? Well, the simplest thing is then to normalize the image to 1. To say, I'm going to whatever the minimum and maximum is, divided by the maximum value. And then every image that I work with, this is only for segmentation purposes. This is not to measure things. Because once we normalize it between the 1 then the absolute values are high. But for segmentation, it would be good to have a normalized set. So what I'd always do is put this node in here. There's nothing to set here. It's just it goes through the image, finds the maximum pixel value, and divides everything. Now, if it's an image where you have a few artificial pixels which are very bright, for example, you label your cells with pixie, and then you have some crystals sitting there, and it's really, really bright. It won't work because then it will get this intensity at the maximum. This might be 10 times brighter than the rest of it, and divides everything by that, so you won't see anything. So this works with homogeneous images, and these nuclear staining was good quality. So this method worked well. So when I run that, what happens is you don't see much. You run it, and then you see an increased intensity, an increased contrast image of the nuclei. Yeah? And this is what I meant. You see that the label is not because of the distribution of DNA. It's not homogeneous. So classical methods do work, but they need some post-processing. So what I did was the following. First of all, I said, okay, I don't need to have the resolution of the stat for nuclear images. They are just nuclei. I just want to find the objects themselves. So instead of using the raw data, I said, okay, let's blur the image by 10 pixels. And you learned about this both on Monday and yesterday. This is what Gaussian blurring does. It makes the object more homogeneous, also, of course, lower resolution. But for large objects where I just want to find them and count them, this actually helps the analysis. So I have that. And then I say, okay, I can now either, as Roman taught you, do some thresholding direct, and you will see this in the when we analyze the cytoplasm, that after the Gaussian blur, we go directly to thresholding. But here, I decided to go with AI-based segmentation. Now, let me explain a little bit about this one. The method we use is called samples, and Roman implemented this into JIPipe, and it has 3 versions: samples 1, 2, and 3. And now also there is samples 4, but a little bit different. So I'm using the latest one in this regard. How does this work? You know about AI, of course, but what happened here, and you may also know about samples, but basically what happened here was that on millions of images of biological data, nuclei, plasma membranes, cytoskeleton, and so on and so forth, they trained a set of 4 deep learning models. And they said, okay, these were annotated images, so they said, okay, these are nuclei. Please learn from these 100,000 images. This is what nuclei look like. These are cell membranes. Learn how to look like. Different cell types. These are cytoskeleton. These are this, these are this. And then they provided the public via publication with these trained models. And if I now take an image of a nucleus and I show it to this model, it will say, oh, this is a nucleus. And it will draw around the nucleus and say, these pixels belong to the mitochondria. Or it says, this is a macrophage based on transmitted light images. And I draw around the macrophage. This assumes that using this node, you provide the node with a model. The model means that certain deep learning frameworks were put together and shown hundreds of thousands of images, in this case nuclei, for example. And then what the model learned is saved into a file, and if you apply this file into our samples node, it will apply this knowledge, so to speak. And when I show it to, when I show an image to it, it will say, this is the nucleus, and I will draw around this for you. So you don't have to do it with classical. So we have to provide a model. And these are called pre-trained models because we didn't train them. The research group who published samples over years. The way it actually works in not the latest version of samples, but earlier versions of samples is that instead of training one model, they train 4 models, different structures of deep learning models. And none of the 4 is always right. They have maybe 70–75% precision of finding that this is the nucleus, this is the macrophage level. But if you let all four of them look at the images and vote, then the combined vote is very precise. So none of them have to be perfect, but they all look at it slightly differently. So the outcome, when they all raise their hands, is very, very precise. And they trained a whole bunch of these models. I will show you what's inside that, and you can see it on your own screen. Please follow through. If you click on that, and then you have this tab here for models. If you click on the triangle, these are the models that they provided us with. Cytoplasm, live cell imaging, live cell 1, 2, 3, 4. We don't really know what the difference is between 1, 2, 3, 4. So what you do is you try and error which model works the best for you. So when I chose cytoplasm 3, even though it's a nucleus, I did this because I tested some of these, not all of them, but those which made sense, and I found that cytoplasm 3 works the best in finding these nicely labeled nuclei. There is actually a nucleus model here, but it didn't work as well as cytoplasm 3. But anyway, so you pick a few of these and you test them. And you say which one works best. And as you see, there's a button here called Add, which means that you can test more than one model. So if I add one more, I could say, okay, I also want to check the nuclear model. And like Ruben mentioned yesterday, with these nodes which accept more than one input, you can avoid having to test every model or every option one by one. You can just set up 10 of them, set it up to run, go have lunch, come back, and it will be done. If one of them, at the end, you don't want all of them, you highlight it and you say, okay, I don't want this one. So I did this when I was working on the project, and I found this is the best one. Now, I'm done with this node. Then what do I do here? If I go to the next node, I have to provide this node with a model. So I added this node here. As you see, it doesn't have an input. It just has an output, and it's connected to the model. The node needs a second input, of course, and that's the image itself. And I connect this one directly to the Gaussian blurred image and put it into here. If you don't do Gaussian blur, you might start seeing pixels which are not identified as nucleus. So these steps here, called pre-processing, they help the AI model to find what it needs to find. Now, what comes out of such an analysis? Well, first of all, if you look through the parameter settings, you have to provide the model with an estimate of how big the objects are. Now, if you don't do this, and there is only one object type in the image, it will work still. But if you had, let's say, depth staining of cells and depth staining of whatever, bacteria, then it will struggle to find which size you are talking about. So then you say, I'm talking about the large objects here. So I set this up, and this is again one of those that you can play with and figure out what this number should be, or you can simply measure in the image how many pixels these objects are that you're looking for. And then very importantly, you have to decide what should come out of this node. So if you click on the triangle, these are the options. It can already draw the region of interest for you. It can draw the flows. I will explain this a little bit. It will show you, or it can show you the segmented objects as labels. Rumen explained what labels are yesterday. And it will also show you, if you want, the probabilities. That is, how likely it is for any given pixel that it belongs to the object that I'm looking for. Now, there are technical details which I won't go into, but you can read up on it on the Cellpose website. For example, how to set up the thresholds. But this is the most important setting. The other thing you should notice is that if your computer has a GPU, you can ask the node, and this is the default setting, to work with the GPU. And as deep learning is always the case, it is much faster if you run it with a GPU than you're running it with the CPU. When you are using the model to estimate or find the objects, it does work quite well with the CPU. When you are training a model, like showing it manually annotated images of nuclei, it must have a GPU, otherwise it takes really forever to run the program. But in this case, the PC has a modest GPU, so you can click this on, and then if you have a GPU, it will use it. If you don't, it won't. So then what do we do? Well, then we simply run this node, and then what happens is it will start the segmentation. Please try that, and it's possible that at this point it will say, I have to install Cellpose 3. Let me know if that's the case in your case. Or does it one?
No,
two?
before another question. In
Yeah.
my case, there's a two-time Cellpose segmentation. Is it necessary to do it one time after another, or is it just one?
So one is enough, but the reason sometimes I do two steps is that the second one will use the output of the first one.
Yeah, exactly.
And then, depending on how good your first segmentation is, it might or might not be enough for that. I sometimes also use actually two Cellpose nodes, one after the other, and I use the output of the first Cellpose node as an input for the second one. You probably prefer. Oh, this is my break indicator. So it's not necessary, but it could be helpful, so it's worth trying. Actually, thresholding is the same, which is the question that sometimes you threshold twice. First, get a raw estimate, and then use that as a mask, as you showed me yesterday to do a second step of thresholding, and it gives you a much better result. So it's up to you, but sometimes it could be useful. Now, before we have a little break, I just want to show you what comes out of this guy. So for each image, we had these 4 outputs. In terms of the region of interest, this already shows you where the nuclei are, and it's doing a very good job, as you can see. It also sees this small one, so we can later exclude that by size, for example, and so on and so forth. It also shows you the nuclei as labels, again, doing a very good job. And this one is an interesting option. It shows you a probability map, and it says that when it is bright, these pixels very likely belong to the nucleus. When it is darker, this is a very simple image, but when it is darker, it's less and less likely to belong to the nucleus. And sometimes, when the segmentation is difficult, I use this output instead of the region of interest or label output, which may not be precise enough yet. And I further process the probability map I can denoise it, sharpen it, whatever, and I feed that either to a classical processing or to a second set of second self-host. Use it as an input. And this works very, very well in many cases. And lastly, what it outputs is that the so-called flow, which is basically indicating that if I see a full rainbow here, just to put it really simple, if I see this full rainbow, this means that the models know what this is. They know this is, in this case, a nuclear. If you have just a little smash of colors, then regardless of what the probability says, it knows, I don't know what it is. Now, for example, we had this case, human macrophages, and the program was like, I don't know what this is. Or I used it to segment electron microscopy images of heat-activated polymer that they used to build these scaffolds in which cells grow. And we were trying to analyze these images, and it said, I don't know what this is. Then you have to train the model further. But since it's a model that was trained on millions of images, it's enough to show it, let's say, 100 images of macrophages, or 100, or maybe even 30 or 50 images of this heat-activated polymer, and it will learn from that. For that, there is a separate node for self-host training, which we don't have time to go into, but it serves exactly this purpose. So, for example, to answer your question, if I'm not happy with segmentation, I could go take the flow or take the probability map, process it a bit, and feed it to a next self-host model. And this will do much better. This is a simple one, but I do this very often. I have two of them, one after. Okay, I will give you guys a few minutes to rest a little bit, and then we continue with the JIPipe as on progress. Okay, let's move on. Okay. So we can, yeah, question. All the samples. Okay. Still, yeah. It's downloading the resources. It's the first time we installed yesterday. But I'll go on and then okay, so a couple of you still have the self-host download, but I move on and then you guys catch up very quickly. It's fast. Yes, so what do we do now? As we saw, there was no need to run a second round of segmentation because the output from the region of interest was already very good. So then we do a couple of filtering. So I did mention that sometimes it will find smaller nuclei because the image was taken so that it took part of the second cell. You don't want that. So the easiest way to deal with that is that you estimate the size of a full nucleus and you say, okay, if I find an object which is much smaller than that, then don't use it. And that's just a filter node, filtering the second two-dimensional regions of interest based on the parameter. If you don't remember the names, I mean, this is obvious, but other names may not be, then edit will list all the options that you can choose from and based on what you can filter. So for later use, I also generated a mask out of the regions of interest. This is basically a binary image where you get 255 pixel values. So if I generate that, then you will see here that these pixels are then 255. So it's an 8-bit binary basically. And we will use this one to calculate later the ring. And then I would like to now generate the perinuclear ring. So for this one, what I will do is the following. I will, first of all, take the nucleus as a region of interest. And there's a node that allows me to make the ROI either larger or smaller. So shrink it or enlarge it. And as I mentioned at the very beginning, after consulting with the biologist, decided that 10 pixels or so in this resolution is where they would define the perinuclear area. So what I do is I enlarge the region of interest, convert it to a mask, and then I do a little mathematics or image or arithmetics. And the reason is the following. If I calculate the mask from the enlarged ROI, then I get the nucleus plus the 10 pixel area around it. So basically this ring here would then be the perinuclear space. So then what do? I take this node called fast arithmetics, and then I take the enlarged nuclei, and I also take the original nuclei as a mask from here, and then basically I subtract this one from this one. And this is this node. And to fill out this node is very, very simple. When you connected the two inputs, they were named I1 and I2, image 1, image 2. And then you say, okay, I want to have the difference between these two. And since we are doing mathematics, let's do it in 32-bit format. So what comes out of this one? Not surprisingly, out comes the area which corresponds to the perinuclear space. As you see, for example, here it also knows that these are two nuclei, not one. So now we have the perinuclear area as a mask. But what we also need for the calculation is the cytoplasm without the nucleus. And how do we get this one? Well, for this one, we have to go to our second workflow, and this is the cytoplasm determination. Now, this one is very, very simple. You take the cytoplasmic image, which was the first channel, channel 0, for example, this one, and you see this is the cell. The dark area is, of course, the nucleus, and we want to measure how much of this fluorescence belongs to the perinuclear space and how much does it belong to the non-nuclear, non-perinuclear cytoplasm. So we take the image, we divide it by the maximum as we did before, and then we run a Gaussian blur on it with a 10 pixel radius. So what do we get from this one? We get a quite nice approximation of where the cell is. Cytoplasm, this is the nucleus, of course, again, various shapes, but they are now fairly reasonably identifiable. So once I have that, I decided, okay, I probably don't need to do deep learning on this one, so I just went with the regular thresholding method, and later we will fine-tune this one, but as a first approximation, this will do fine. We will remove these small areas, we will fill in this hole, but as a first approximation, this works well. Now, for example, for the ladder, where we had these little holes inside the cells, if we run this node, which is called morphological hole filling, then you will see that the hole that used to be here, now it's gone. So we have an approximation of the cytoplasm, and then on the one hand, we can export this one, and on the other hand, we can also now calculate those images where we have the cell itself minus the nucleus and minus the perinuclear space. So what do we output from here? Basically, can I shrink this one? Yeah. Basically, now you have to decide what you need to pass on. Yep.
So how long it takes to download the samples or is
It is for it's just if the network is very slow, then it takes
like 15 minutes now?
Yeah, it could be. So just follow, and then when it's done, just run it. Are you here for the exam? Are you over? No, not then. We'll teach you that.
Yeah.
And you can just pay attention if you're gonna do right.
Of course, that there's issues with the
Yeah,
update.
yeah.
Of course, when our course is not any other.
So sometimes it can take a while. Okay. The output, and here I wanted to explain something that not too long ago, Ruben decided very wisely that there should be the possibility of having more than one output node from a compartment. And that was when he saw me generating outputs like that, or even worse, which are like a kilometer long, with a million outputs, because I need them, but I don't necessarily need all of them in all connections from this compartment. Now, this is a simple situation, because if you remember our compartment view, it's a linear thing. So from here I go there to I go there. So I only need to pass the information to one additional compartment. So this means that I can work with one output. But if I had parallel paths, and if I have a bit of time, I'll show you with the other project, then it might be wise to have more than one output and design each output depending on where it is going to. And as you remember from yesterday, the goal or the purpose of the output node is to pass the information from the previous compartment to the next one. So this means that what you put here is what you are going to need later. You do not put things out which you don't need. For example, this node is not outputted, this node is not outputted, because I don't need them. What I do need is where my perinuclear ring is, where is the extended nucleus, the nucleus, and so on and so on. So you connect the outputs that you need. Again, like it was explained yesterday, it's by far best if you just connect new outputs to the plus sign. It will open a conversation tab, will ask you to name it, but it will detect the type of the data automatically. And I have seen this yesterday. Some of you made mistakes of trying to name it manually, and you make a mistake, and then JIPipe complains that I cannot connect this data type to the other. You don't have to know the data type. That's how Ruben designed it, that it figures it out. Sometimes you want to change it yourself because you want to go with a different bit depth, for example. But in general, it's by far the simplest if you just connect to the plus sign. So what I did was I designed those outputs which I needed for the next step. Now, what is the next step? Well, now we have to deal with the quantification task. So if I go to this one, what you will see up here, of course, is the output from the previous node. And then what we will do is we divide the processing into 3 steps. And you can, if you want, build 3 different compartments here. And you say, okay, one is designed for the ring, one is for the cytoplasm, one is for the nucleus plus the ring. It's perfectly valid. In this case, what you would do is design 3 outputs from the previous compartment, each designed for its oncoming sequential compartment. But it's not terribly complicated yet. So these are basically 3 very comparable parallel processing paths. So I did more pipes. So I did not put them into separate compartments. But if you are a fan of simplicity, then you may want to do that. One very useful tool, which I think you learned about yesterday, was these text boxes, which don't do anything at all. They just visualization tools where you can draw a box around the nodes which carry out a certain function. You do these with that icon there, and then you can name it, you can decide on the color, you can decide on the size of the font, and I find them extremely useful because then when I look back at it, especially a couple of months from now, I'm like, okay, what the heck did this path do? And this is also a couple of months also when I was preparing it for you guys, it helped me really that it says this is that, this is that, this is that. So use it accordingly. Now, what do we do for these? So first of all, we can process the ring. And for this one, first of all, we need to assign them as objects. So objects that can later then be measured, and we say, okay, for each image, there is a cell, and for each cell, there is a perinuclear ring. And this is done by assigning these pixels into a component, connect them into one component. And this is then carried out by this node called connected components label. As the name indicates, you get the input from the ring output over here, connected there. But the output of this one will be a label. So input was an image, in case binary, but the output is a label. What is a label? This was explained yesterday. A label means that each object that you identify in the image, within the object, each pixel carries the exact same value. Each pixel is set to 1, 2, or 3, or 4. This means that those pixels which carry the value of 1 belong to the same object. Those which carry the value of 2 belong to another object. But this way, segmentation becomes much easier. So let's run it, and then let's see what we get out of this one. So we run it, and make this one a little bit bigger. So now we have the perinuclear areas identified as objects, and as you see now, it knew that these were 2 different regions of interest, but it now converted to labels, which means that, as Rumen explained, it can have a hole. So the label itself is now this ring. A region of interest cannot have a hole in it. So we have to work with labels. And as you see, the colors of these 2 rings is different. This one carries the value of 2, and this one carries the value of 1. If you can see it from back there, this is 1, and the other one has the value of 2. So these are 2 different objects. And now it's very simple to keep track of them by just monitoring the value. Those which only have 1 only carry 1 value. Okay, then what do we want to do? Well, we want to measure, or that was the request from the biologist. We want to measure the characteristics of the image in these perinuclear rings. So I use a node called extract label statistic. This means it needs an input where I connect the labels where I want to measure, and it needs an image from which I want to measure. So what do I need for that? Well, for this one, I need the output from my labels. This is filtered by area. What goes here? And then I mentioned it to you that the output on which I measure is the raw data. So basically, this is the image which came directly from the diff file and contains the first channel 0. So then what does this node do? It will take the labels, apply them on this image and measures whatever you select here. And what I want it to have is the area, the location, and the pixel values. I did not want the rest of them. So what you do is, up here you can either select all of them or you can select none of them. And if you select none of them, then you can pick a few that you want to measure. And this I would like to emphasize because there are like 30 parameters that ImageJ measures by definition. And this will slow your workflow tremendously down. But especially the morphological measurement. So it measures the circularity, ellipticity, aspect ratio, blah, blah, blah. For this one, it has to go to every pixel of every label and measure the shape point. So this takes a long time. And if you are working on a lower capacity PC or any PC, if you don't need it, don't calculate it. The default is that these guys, when you drop this node, these guys are all clicked. So keep an eye on it. One trick is that these guys are typically hidden. So when you open the node, it's going to look like that. And all these parameters are hiding here. So if you click on that, then you see them all. And what you will see is all of them clicked. And just make a smart decision. And this one, measured in physical units, keep an eye on that because you have to be consistent. It's perfectly fine to measure distances or areas in pixels. But then you have to know that it's measured in pixels and it has to be measured or should be measured in pixels throughout the workflow. Or you can measure it in square markers. But for that, you have to be sure that your resolution, your pixel size is set up correctly. If you load a TIFF file which is not calibrated, it will assume 1 pixel per 1 micrometer or 1 micron. In this case, you remember earlier this morning, it didn't know that it was 54 nanometers pixel size. If you are sure about that, then you can measure in physical units, otherwise it measures in pixels. And it's perfectly fine, because you can always convert one to the other as long as you're sure what these sizes actually are. Okay, so now we had these numbers out. Let's start saving the results. Let me zoom in a little bit here. We get the output. Let me run it. So for each image, we have the area of the ring, the mean fluorescence, standard deviation, and the location of the ring itself. I calculate also the median. Oftentimes, if the median is very close to the mean, that means that your number of outliers is not very large. So this means that this is a fairly homogeneous distribution. And this is the first label. There is only one nucleus in this image. Now, when you want to save this data out, you do this with the export data tab. Sometimes you want to make the name of your output a little bit more meaningful. And if you look at here, when I open this again, it said area mean. These are all meaningful, but it says label ID 2. At the end of the analysis, I would like to measure all these parameters for the perinuclear ring the nucleus plus the perinuclear, and the cytoplasm without the perinuclear. So there will be 3 measured eggs plus the whole cell, so 4. And at the end, I would like to make 1 table like this, because if I give the biologists 4 tables, they won't like it. But this means that somewhere here, I should carry the information that this is a measurement from the ring. And if I save the label ID, for example, it doesn't say, is this from the ring, is this from the cell, is this from the nucleus? So what I do is I apply this node and I say, the column that used to be called label ID, let's rename this one and put the origin of the object. Label ID ring. And now when I later merge these parallel paths into 1 table, I will know where these numbers came from. And you can do this with this one. And then all we have to do is, in this case, export the data. And now the exported data will have the identification, then this is from the ring. So we run this one. It goes through, and then it goes to the analysis folder. Now, how do I know where this one is going to? Well, that's why we introduced at the beginning the analysis folder user path here. So I built a name, and this is what you guys learned yesterday, that the saving can be and should be automated. Fully automated. It is possible that you run your results and you save them in the cache, and then from the cache, oh, it's lunchtime. Thank you. And then you manually save them from the cache. There's nothing wrong with that, but if you have multiple data sets, then it's gonna hurt you or time schedule. So let's automate this one. You learned about this yesterday. That the reason I asked you guys to set up this project data directory was that I can now use it. If you remember at the beginning of the workflow, we used the data folder user path. Now I'm going to use the analysis folder user path. So instead of having to type in here c/Zoltán/teaching/blah, blah, blah, I just use the project data directory called analysis folder. So this is the function call, and this is then in square brackets the name of the user-defined path. Very, very simple, and you never have to change it again. So as you carry on building the workflow, your saving expression will always be the same. It's very, very simple and much, much faster to build. And then you add to that whatever you want. So for example, you want to add the name of the subfolder, and you want to add the name of the image, and you want to say, well, this data is coming from the ring. But you will also notice now that instead of plus subfolder 2 plus image, instead of all this, I could have just said image 2, because that's the combination of the subfolder and the image name, which we introduced at the very beginning of it. So here what I do is I use that new annotation, if you remember it, and I precede it with the second subfolder. So now I know not only the name of the donor, but also, for example, when the experiment was done. So I merged the names of 2 subfolders. And this will be then the name of the file under which this data will be stored. And what type of data it is, it will figure out based on the input data. So when I run the workflow, it will save the results automatically. So if I go here, and then I go to analysis, and then I go here, then this is the data, 1107. So this was saved a few minutes ago, fully automatically. So I know the name of the experiment or the condition. I know the name of the donor, and I know the name of the image, and I also know where it is coming from, and it's a CSV file. This is fully automated, and this means also that when you run the project, you don't have to worry about forgetting to do that. Now, when I save these results, I can also, as an option, add, what am I doing here? I can also add additional information to the table itself. So now I saved the intensity, the area, blah, blah, blah. But maybe I would also like in the table to have the information about the image name and the condition. So when I give this to the biologist, he will know from which conditions these numbers came from, so he can tabulate it, make a nice bar graph, etc. And for this one, we have a node called add annotations as columns. And it does exactly that. For example, it takes the second layer of subfolders and adds it as a column in the table. Or in the second one, it adds the image name as a column. So when I run this one, it will add the name of the image. So when I run these guys and I look at the table, I have two more columns. I have subfolder and I have image. So now when I save this one, I have all the information that I need. So now this can go to the export node, and then we are done saving the results. Now, if I want to also demonstrate what this looks like, what I can also do is visualize what this perinuclear ring looks like. And if you remember, they were like rings, just like that, yeah? But if I want to see the edges, for example, as a nice ring, then I can use this node, which is called get label boundaries. And it does exactly that. It translates the label into its inside and outside perimeter. Why do I do that? Well, in this case, I just do this specifically to be able to visualize my output. And for the visualization, I will use the blend image node. You learned about this yesterday. I define which image should be at the bottom. This should be, in this case, the label image itself. And then the top should be these label outlines. So when I run this one, what I will get is an overlay of the images. So if I zoom into this one, then we have the fluorescence image overlaid with the perinuclear space. I made these lines a little bit thicker so you see them better. And this was done with a very simple node called dilate labels. And I set the value to 3 pixels. Okay, so in this case, I now have my overlay image. I added a display contrast increment, but basically now what we have is an image, come on now, where you can overlay the cell with the perinuclear space. And in order to give this or keep it for yourself as a reference or give it to somebody at the biology department, you can export this image. And let's do this also automatically. So once again, we do the exact same principle here. We use the project directory code analysis folder, and we build another name. It knows what it is. I define what image format it should be. It should be a PNG in this case. And I build up the name exactly the same way as I did. And again, the advantage of this is that to build this string takes really a simple copy and paste. And all I had to change was this overlay signal to get the output automatically done. I didn't have to figure out which folder it should go to and what the folder name is. Very good. So now, in a parallel path, this is really just for fun. I also aligned the perinuclear space with the nucleus itself. So what you will see here is, if I zoom in here a bit, this is to show that actually we can be happy with ourselves because we did a very good job. So the perinuclear segmentation really outdrew the nucleus perfectly. Remember, this was done with the cell pulse AI-based system. If you do this based on fluorescence, you get good results, but not necessarily as perfect as this one. So here you see where your perinuclear signal comes from, and these images are saved as a reference for later when you overview your experimental outputs. So we are done with the ring, and now we turn to the cytoplasmic analysis. This is really very similar. What I will do is, again, I will turn these cells into labels, but then these little ones, which were artifacts, I don't need them. So the simplest way to get rid of it is knowing that the cell is by far the biggest label in this image. I can use this simple little node called keep the largest label. There's nothing really to set here. You just put it in, and what it does is it removes all the labels except the biggest one. So now we have one cell per image. And this solves exactly or produces exactly what I need to know. And then what do? I do the exact same I did with the ring. I get the statistics. Once again, remember to set these guys up. I actually just copy pasted this node here. We get the area, the center position, and the intensities. Again, I'm measuring in physical units. Once again, you have to be consistent, otherwise eventually you will get confused when somebody asks, okay, is this in microns or is this in pixels? Well, I don't know. So be consistent. I renamed the column again from label ID to label ID cytoplasm. Honestly, these are copy-paste nodes where I just change what I need to change. I export the data. I again build the name the exact same way I did before. And you see how fast this goes now, yeah? Because the whole thing is reproducible. Add the folder name, and I add the image name. And now I want to have a merged table. So if I run it up to here, I have 10 tables. Each table represents one image, cytoplasm area, blah, blah, blah. We have the folder name, we have the image name. But what if I want to have this in one table? Well, then what you can do is merge these rows into one. And then what we have is a summarized table. And this is now good enough to be used for analysis and making bar plots. You know where the data is coming from, you have all these parameters, but also, because we were clever, we also kept track of where these numbers are coming from. So instead of just having a table of 10 lines, we know exactly where they are from. And when we run this for various subfolders, I can use, this is useless, this column, because it's just a 1, 2, 3, 4. But this is useful. This I can use to group my data, and then you do those nice bar plots that biologists very nicely do, where you show that under such and such condition, the measured value was this, and under the other condition, it was significantly smaller or larger. With this very simple node, then you merge these guys together. And here, if you look at this Roman explained yesterday, this is a very simple way to do this. If you go to the inputs table, which he explained, by setting this to all into one batch, we'll take care of merging all of these inputs into one table. And as a result, like I said, you only have one table now. And then you export it. The exporting again is done with the exact same way. You build up the path of the name of the file, and then you do the exact same as we did before. We get the edge, dilate them, and then we calculate the overlay images. Now we are just repeating what we did before. And then what you have is an outline of the cells together with the periphery. This is the nucleus, this is the outline we got, and then the fluorescent signal. So we are doing a pretty good job here. And then after contrast adjustment and rendering to RGB, this makes it a bit faster. We export the images the exact same way as we did before. And then finally, we measure the area for the nucleus plus the ring. The exact same path. I use the output from the previous node, that is nucleus plus the ring. Filter them by size. So if I run this one, this is the nucleus together with the perinuclear area. This is something they wanted to measure together. So this is larger than the nucleus by 10 pixels. And then we go through, get the statistics, rename the column. It's really the same that we did before. Export it. Then we add the name of the subfolder, add the name of the image to the table. We merge the table rows. We set the input the same way, all into one batch. And if you run this one, we get a table where we have the numbers for the nucleus plus the ring, keeping track of the folder and the image name. And then we save it. Then we get the outline, dilate the labels, blend the images, all the way through the same thing. And then we have our overlay images where we show the outline of the nucleus plus the perinuclear overlaid over the image. We save it. So what we did now was we documented each and every step of the intermediate results, plus at the end we have the numbers saved in a smart table. Now if you also want to merge all of these tables into one, then what you can do is take the input from all these other tables. We had this one, this one, this one, where we merge the rows, and we take these tables and then we merge them together. And then what we have is the cytoplasm, the nucleus plus ring, the ring, and the entire cell, all together in one table. So the different regions are separated per column. The different images and conditions are per row. So at the end, what we have is one big table. And when you provide this to plotting, or if you are a bioimage analysis like we are, and you give this to the biologists, they are really happy, because then they get one table. And then in one table, you have everything. All the conditions, all the regions that you wanted to measure, so they can go into Prism and plot bar graphs. So this is our final output. When you do your own post-processing or post-calculations, you can, of course, do as much as you want in JIPipe, but keep in mind at the same time that JIPipe is not designed for very high level of plotting or very high level statistics for further processing. This you can do in Python, for example. If your output is, so usually I think my output as CSV files. Simply, I find it's easier or simpler to work in Python. But you also learned yesterday how to do the Excel, which is also very useful. But if you have your output in CSV, then you can, for example, write a Python node, Python script, which takes the CSV files and then puts them into fancier plots or calculates statistics. So after a certain point, it's not worth putting these levels of activities into JIPipe. It might be slower than doing it in Python, less flexible. So you have to be smart about where you start with the Python, where you start with the JIPipe level of processing, and where you start with Python. And this really saves you time, because if adding the statistics and plotting and whatever to JIPipe increases your runtime by a factor of 2, so instead of taking 2 days, it takes a week, then it's really not worth it. Then it's much better to save CSV, put a Python script together. And then what you do with the Python script is again up to you. You can put, as Roman explained, there are Python nodes in JIPipe where you can use either native as a script or Jython, depending on how you write your code. That works fine. I use this not in this project, but sometimes. Or you can just keep it separate. As long as your naming of the CSV file is consistent, which it is, because we are using automated saving and automated naming, Python will have a very easy time to find them. We can use also Python agents, AI agents like we do. We want to set this up to, if you use root code, for example, where you tell the agent, okay, I have CSV files by such and such naming structure. Please go in, find the column for area, find the column for whatever, and then group the area value based on the origin, and then make a plot about it. And then the agent will do this for you. So this is the practice I follow most of the time, that at some point I stop and then go, and these Python scripts can also be shared with biologists and they can run it. So it's really not a big deal these days to get this thing done. In terms of plotting and three-dimensional surfaces, we don't really have that yet in JIPipe. We have students working on some parts of it. We also don't have the convolution yet in JIPipe. I supervise a student together with Ruben, who is doing that, hopefully soon. Plotting, we have a student who's working on Python library-based, fancier two-dimensional, three-dimensional plots. At the moment, we don't really have them. We have some basic libraries based on Java, which will do good enough plots so we see what the output is, but these are not publication output by any means. We don't have 3D surface rendering. We use Humaris for that. You can also use open source tools. We hope to find somebody to write this for us. We don't have time for everything. But in terms of processing, automating up to this point, it is a very, very useful tool. So what I recommend is that you go through what you learned and use it, because you will forget it if you don't. So passive, if you become passive knowledge, and then you have to start again. So I recommend that even those of you who didn't get through the samples, download it, run this in the evening, go through the notes, learn how this is built, because this will help you able to use it and then use it. For those of you who do the homework, what you learned, I hope will help you, because the task really will be doing a complete JIPipe workflow with multiple compartments, I think 4 compartments. So try to follow what you learned today to build those workflows. So as far as I'm concerned, I'm done. We still have a little bit of time. If you guys, I hope if you have like half an hour, if you guys have questions, then we are all happy to answer. And after lunch, we start the homework. For those of you who want to stay, you are very welcome. The homework is now here. These are the instructions. You can take one each. It will explain exactly what to do. It contains the microscopy. In the microscopy question, there is one question, question 7, where at my lecture we didn't get to all the aspects. So either look them up or you can simply ignore question 7. It's up to you. It's easy to look it up on Google. I will not count that as a problem if you don't solve it because I didn't have time to go through all of them, like second harmonic generation and so on. And then you will have this detailed description of the JIPipe class. Quite involved task, I would say. But don't forget about the microscopy either because you have to pass both of those tasks. And the instructions are there where to send how to as soon as you are done, please submit it so that Ruben and I can go through them. You have until tomorrow at 8 o'clock.
8 o'clock the evening.
8 o'clock in the evening. In terms of the room, we open it at what time tomorrow, Ruben? 10 or 9 or
As early as possible.
so
You will make.
So we open it at 9 for example tomorrow and you can
Maybe